discussion period.
So, John, is that all you got?
Can I have the slides back up? I've got another 20 or so on this.
Now, this is a very provocative presentation, and Judy made a very provocative presentation as well. So we get down to basically where are we? And I want to go back to the premise of this session. I think first of all, the good part about this whole meeting is that I think there's acceptance that there's something real that we need to find. The debate comes in whether or not we have found it as yet. But if we've made that first step, that's a big thing, and then you can plan from there. So we're left with, is this an infectious agent? Probably a virus. It has those characteristics. And if it is an infectious agent, is it a single agent? And today we've heard that it might be EBV related, that EBV can reactivate under stress or other conditions and could cause a syndrome similar to this through cytokine production in monocytes or other cells. We've heard that it might be an enterovirus. The whole class of enteroviruses might be the inducing agent to some common cytokine pathway that results in these symptoms. And then we've heard that it might be XMRV, or XMRV like murine leukemia viruses. And then we heard that it might not be XMRV, that XMRV could be a contaminant, and that it could have happened from a recombination in these through the nude mice of an original prostate cell line. And lastly, I want to lay out the other postulate that I had was that whatever the etiology of chronic ME/CFS, that it happens in a subset of patients, so not in all patients who get any virus or multiple viruses. So there must be something unique in the host that facilitates the syndrome, and I don't think that has been sufficiently explored. So that's another outcome of this meeting. So, yes?
What do you mean by it happens in some? What's the it that you're talking about?
Well, the syndrome that we call ME/CSF occurs in only a small minority of people who have a viral infection. And whether it's a particular virus infection, even there, and that's classic for viral infections. Some people get sick, some people don't get sick, some people get over it quickly, some become chronic carriers. But here it seems to be a very small piece of the pie who go on to this full syndrome, and what is it about those people that makes that happen? So I think those are some of the outcomes. So now to get down to the nitty-gritty, I want to open this up to questions for any of the four speakers. Yes?
This last point that you made is a good point. The last point you make is a very good point. I didn't get a chance to mention this, but here's something that fits within this context. It turns out that with patients with different kinds of EBV-associated disease have different antibody patterns. Nobody has never understood why that's the case. People who have Burkitt's lymphoma have one pattern, a person who has NPC has another pattern, infectious mono has another pattern. We don't know why that's the case. Could that, whatever's behind all that, determining who actually shows clinical symptoms, be related to this issue as well? We just have never resolved that issue.
Thank you. Nancy?
As someone who's not a retrovirologist, I'm always, the discussions are fascinating, and that was a tremendously elegant detective work. But I'm left with two things. Why would someone make antibody if there's not an antigen? That really bugs me. And then the other one is, I don't know how these were run, and I imagine if things were in batches, you could get difference between controls and subjects. But if they were truly randomized or in the same runs, you should see the same kind of stuff. So data showed the controls equally positive or more, yeah, that made sense. There's a contaminated set of data, but these other data that we're seeing in these publications looks pretty clean and separate.
Well, in the Lowe et al. Study, the controls and the samples came from Tony's lab were taken many years ago. The controls were fairly recently collected samples from the NIH blood bank, if I remember correctly. So they were clearly not matched and processed the same. And
No, that doesn't make sense, because the old samples would be the ones you'd want to test. The ancient samples, pre-1992 samples, are the ones you'd want to test. And his samples are older samples in that cohort. So I don't get that either. I mean, sure, they're not identically processed, but if they were run in the same lab and the same assay systems at the same time.
well, but there would have been differences along the way by which they would have been collected. They would have been collected in different kinds of vessels, some in tubes, some with EDTA or heparin or whatever. I mean, any of these, heparin, for example, is probably a very good possibility for contaminating mouse DNAs. Unless you start out with a study that, because of this problem, that such tiny, you've seen my slide with my swimming pool, such tiny amounts of mouse DNA can give rise to this kind of detection. Unless you start out with using exactly the same tubes, all of the same chemicals, exactly the same reagents, starting from the time that you put a needle into somebody's arm to collect the blood, the possibility exists that you'll have a batch of tubes that have been contaminated along the way. You'll have some reagent that was used in the immediate collection that was contaminated along the way. The only way to do this, and studies are now being set up, and that will do exactly this, and we'll go back and revisit the Ian Lipkin study, for example, is being set up to ensure that exactly the same reagents are used all along, and supplies are used all along the way. And it's the only way to be really confident that this is in fact the way it's working. I can't speak for Judy's sample. I just don't know enough about how they were collected, but it was very clear. And I promise not to talk about antibodies. But I will point out that if you go back to the very old AIDS literature, you'll find antibody studies that showed HTLV1, that showed this and that and the other thing that can't possibly be right. I mean, the anti-HIV test, the HIV tests have a very high false positive rate, for example. That's why you need to use two of them to get a reasonable level of specificity. But I can't address the antibody observations. It's not what we've been working on, and that needs to be worked out. But all I can say is that, and it's possible. I'm not going to deny the possibility that there is, in fact, an antigenically cross-reactive virus out there that may be infecting people. That's not XMRV, but cross reacts antigenically. These antibody tests are very broadly reactive to things that are react well with things. Judy mentioned friend virus. Friend virus is way off that tree on a very distant branch. Most of the PCR reactions would not detect that virus because it is so distant. And there may be a virus like that in these people
there clearly is a human gamma retrovirus in these people.
I'm not going to deny that as a possibility.
We measured viral proteins throughout the study in the science paper, and we used things that were cross-reactive. So it was only the definition, and this is the first time it's ever happened in human retrovirology, that we defined a virus based on a single sequence that he calls XMRV. I mean, we call XMRV a xenotropic MLV-related virus. That means any xenotropic virus that is infecting human cells. As I mentioned, DG75 is a specific case in point. That's another virus that wouldn't react with any of these tests except our antibody test. It's a human gamma retrovirus. It's very closely related but not identical.
No, that's not true, Judy. It would have easily reacted with the viruses with a test that we've used at NCI Frederick.
It didn't react with the primer pairs we used in Urusmanol or Lombardi at all.
I think that's correct, and those primer pairs.
So there's another family member there. It reacts with our antibodies in a Western format.
The silver